Hello friends, welcome to some biology class. Friends, today we shall discuss about bacterial cytoplasm. Bacterial cytoplasm is a gel-like matrix which, like that of eukaryotic cell, is separated and protected from the external environment by the plasma membrane. It usually lacks membrane-bound organelles. Water accounts for 70%. The bacterial cytoplasm contains different types of inclusion bodies, ribosomes, nucleoid, and plasmids. Previously, it was thought that cytoskeletal elements are absent in the prokaryotic cell, but recent evidences indicate that cytoskeletal elements are also present in bacteria, which are structurally and functionally similar to their eukaryotic counterpart. The plasma membrane and everything within it is called as protoplasm. Thus, the cytoplasm is a major part of the protoplasm. This figure shows the structure of a bacterial cell. The cell is protected by three layers. Outermost layer is the capsule. Inner to the capsule there is cell wall, and beneath cell wall there is plasma membrane. This is the gel-like matrix known as cytoplasm. Already you know that in bacteria a membrane-bound nucleus is absent, and the genetic material is located in a irregularly shaped region known as nucleoid. The cytoplasm also contains numerous ribosomes, which are the site of protein synthesis. Bacterial ribosomes are of 70S type. The cytoplasm also contains different type of storage granules, which are known as inclusion bodies. In addition to this, the bacterial cytoplasm also contains different enzymes, inorganic and organic nutrients. On the surface of the bacterial cell, these are the ultra-thin appendages known as pili or fimbriae. Many species of bacteria are motile. They possess whip-like locomotory appendages known as flagellum. So friends, this is about the structure of a bacterial cell. First, we shall discuss about inclusion bodies. Inclusion bodies are the storage granules of inorganic or organic substances. They store carbon compounds or inorganic compounds. Some of the inclusion bodies lie free in the cytoplasm. Example, polyphosphate granules, cyanophycin granules, and some glycogen granules. Remaining inclusion bodies are enclosed by a single layered cell of thickness 2 to 4 nanometer. Examples of enclosed inclusion bodies are poly-beta hydroxybutyrate granules, some glycogen and sulfur granules, carboxysome, and gas vacuoles. Organic inclusion bodies usually contain either glycogen or poly-beta hydroxybutyrate. Glycogen is a branched polymer of alpha-D-glucose. In a glycogen molecule, a large number of glucose units are linked together by alpha-1,4 and alpha-1,6 glycosidic bonds. Poly-beta hydroxybutyrate granules contain beta hydroxybutyrate molecules which are linked together by ester bonds between the carboxyl and hydroxyl group of adjacent molecules. Cyanobacteria and other photosynthetic bacteria contain two distinctive organic inclusion bodies. They are cyanophycin granules and carboxysome. Cyanophysin granules are composed of large polypeptides which contain approximately equal amount of arginine and aspartic acid. Cyanobacteria and other carbon dioxide fixing bacteria contain another type of organic inclusion body known as carboxysomes, which is polyhedral. This granule contains the enzyme ribulose 1,5-bisphosphate carboxylase oxygenase, which is also known as Rubisco. This enzyme is critical for carbon dioxide fixation. A remarkable example of organic inclusion body is gas vacuoles that provide buoyancy to some aquatic prokaryotes. They are present in many photosynthetic bacteria and some other aquatic prokaryotes. Gas vacuoles are the aggregates of enormous number of small, hollow and cylindrical structure called as gas vesicles. Prokaryotes with gas vacuoles regulate their buoyancy so as to float at a depth necessary for proper light intensity, oxygen concentration and nutrient level. Prokaryotes with gas vacuoles descend by collapsing the gas vesicles and float upward by constructing the new ones. Two examples of inorganic inclusion bodies are polyphosphate granules and sulfur granules. Polyphosphate granules are also called as volutin granules. They store phosphorus in the form of polyphosphate. Polyphosphate is a linear polymer of orthophosphate linked together by ester bonds. These granules are also called as metachromatic granules because they show the metachromatic effect. Another inorganic granule is sulfur granule. Many prokaryotes use sulfur granules to store sulfur temporarily. For example, photosynthetic bacteria can use hydrogen sulfide as photosynthetic electron donor and accumulate the resulting sulfur either in the periplasmic space or in the special cytoplasmic globules. Inorganic inclusion bodies can be used for purposes other than storage. An important example is magnetosome. These inclusions are used by aquatic magnetotactic bacteria to orient themselves in Earth's magnetic field. Magnetosomes are intracellular chains of magnetite particles. When examined with electron microscope, the bacterial cytoplasm is seen as packed with ribosomes. Ribosomes are very complex structure. They are composed of RNA and protein. They are the site of protein synthesis. The cytoplasmic ribosomes synthesize protein which are destined to function within the cell, whereas the plasma membrane associated ribosomes synthesize protein for transport to the outside. In bacteria, ribosomes are of 70S type, which are composed of a 50S and a 30S subunit. Next important structure is nucleoid. One of the most important difference between prokaryote and eukaryote is the manner in which their genetic material is packaged. Eukaryotes have two or more chromosomes that are contained within a membrane-bound nucleus, whereas in bacteria, a membrane-bound nucleus is absent, and the genetic material is located in a irregularly shaped region known as nucleoid. Nucleoid consists of 60% DNA, 30% RNA and 10% protein by weight. Most of the prokaryotes have a single circular double-stranded DNA molecule. Some other bacteria have a linear chromosome, whereas few others such as Vibrio cholera have more than one chromosomes. In bacteria, the genetic material is efficiently packaged so as to fit into the nucleoid. The genomic DNA is extensively looped and coiled, probably with the help of RNA and some nucleoid proteins such as condensin to form a specific structure. In addition to their genomic DNA, bacterial cytoplasm also contain extrachromosomal DNA molecules known as plasmids. Plasmids can be circular or linear. They are autonomously replicating. Some plasmids can also integrate into the bacterial chromosome and are called as episomes. Some plasmids produce only one copy per host cell, whereas others produce more copies. Some plasmids contain genes for the construction of hair-like appendages known as villi and transfer copies of themselves to other recipient bacterial cell during the process of conjugation. One of the best studied example of conjugative plasmid is the F plasmid or fertility factor of E. coli. Some conjugative plasmid also contain genes for antibiotic resistance. Such plasmids confer antibiotic resistance to the host cell that contain them. These plasmids are called as R plasmids or resistance factor. In addition to these, some other types of plasmids have also been discovered. They are bacteriocin encoding plasmids, virulence plasmids, and metabolic plasmids. Bacteriocin encoding plasmids contain genes for the synthesis of bacteriocin. Bacteriocins are bacterial proteins that destroy other closely related bacteria. Cole plasmid contain genes for the synthesis of colicins. Colicin is a type of bacteriocin that is produced by and directed against other strains of E. coli. Some other bacteria contain plasmids that codes for the synthesis of bacteriocins, which are effective against other species. Another type of plasmid is virulence plasmids. Virulence plasmid encode factors that make the host more pathogenic. In addition to this, some plasmid contain genes for the synthesis of enzymes which degrade aromatic compounds such as toluene, herbicides, and sugars. So friends, this is all about bacterial cytoplasm. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much. Good night.